So, here's where we're going to start to get into metabolism and nutrition, right? As it says right there on the top. So if you weren't sure, all you had to do is read it. But we all have heard about metabolism. My God, we've all heard about nutrition. Oh man, you can't eat this, or you should eat this, or you shouldn't eat that, or oh man, carbs are the devil, or oh, fats are evil, eggs, oh my God, don't touch those things, or wait, what? You gotta eat whole grains, or I mean, there's so much information out there. It's hard to know what's legit, what's not, what should I pay attention to, what shouldn't I? And that's the thing, right? A lot of it is correct, and a lot of it is incorrect, right? For instance, if somebody came into my office and said, "Hey, I want to lose weight," I would tell them, I would tell them X, Y, and Z. I would tell them certain things to do, certain things they shouldn't do. Then somebody comes in an hour later, says, "Hey, I want to put on muscle." I would be telling them things that were completely contradictory to what I had just talked about to the person an hour before that, right? Because their goals are entirely different, right? Trying to, for instance, decrease adiposity is entirely different than trying to increase muscle mass, right? And all of it's dictated by the hormones that are present. All of it's dictated by the nutrients you put inside of your diet and by the metabolism that's actually occurring inside of your body. Right, so we're gonna start to look at kind of what some of these different steps are, right? But when we think about it, like, all right, well, why is it we eat? Okay, because food tastes delicious, right? I don't know. I didn't need to have like thirds tonight for dinner, but I did because it was delicious and I ate it, right? But why, right? Why is it I went back for thirds? Well, all right, that's because that might have been a little excessive, but just eating in general, right? The different nutrients, right? Like I had mentioned, the different chemical processes inside of your body, they require different nutrients, right? So just like you could put gasoline in your car, you put oil in your car, right? Your car utilizes that gasoline, burns the gasoline, and uses that pent-up chemical energy inside of the fuel to create mechanical energy. That's actually exactly what we do as humans, right? We take the chemical energy that's pent up inside of those chemical bonds within the nutrients that we consume. Well, actually, we utilize the nutrients to create this chemical energy in the form of ATP, on which we then render that energy from the ATP and create mechanical energy, like we lift things with it, right? So, again, same thing right here. Not going to belabor the point. That's what we just talked about. So there's two different types of reactions. You would have talked about this at first back in like chapter 2, which at this point would have been sometime, assuming you took 121 in the fall, it would have been somewhere right around like the end of August, right? So we're talking quite a while ago. But there's two different types that we're going to talk about: catabolic and anabolic. Catabolic reactions, you see breakdown of complex molecules, and this releases energy. Cool. So this is where I take a bite of my cheeseburger. I take a big sip of milkshake, vanilla. Strawberries okay as well. Fresh bananas even better. No chocolate shakes. Get that crap out of here. Anyways, catabolic shakes. Catabolic reactions. That's, again, taking those large molecules and breaking them down. Right, so just like I take a bite of a cheeseburger and I break it down to smaller and smaller and smaller molecules, same exact, that's a catabolic reaction. Just when we talk about it inside of the body, molecules that are already small, right, but making them smaller, like a triglyceride to a fatty acid, that's a catabolic reaction. Right. Anabolic is the opposite, the building of complex molecules. So this is where we then take, for instance, amino acids and convert them into proteins, or we build them together to create more complex proteins. These two reactions, this is metabolism. Right, think about it. The food I consumed, I had to break it down in order to be able to absorb it. Right, just like the last class, using the example of the ice cube. Ice cube wouldn't fit through the screen. I had to break it down by smashing it with a hammer, and then I had to dump salt all over it. Those two things help to melt and break down the ice so it then fit through the screen. Again, those are catabolic reactions to think about. Well, then all those ice, those water droplets fall through the screen, and if I collected them in a dish. Now, an anabolic reaction would be, well, what if I wanted to refreeze those water droplets? All those water droplets are all separate. What if I wanted to combine them all together and refreeze them and turn them into a giant ice cube again? That's an anabolic reaction. I'm taking those small droplets and combining them into a larger and larger ice cube, for instance. Amino acids in the proteins, right? Your muscle. It's not like you eat a steak, eat a burger, eat chicken, whatever it is. You don't just eat that and all of a sudden that lean muscle mass just somehow makes its way through the intestinal wall and plants itself into your bicep. No. Right? Through catabolic reactions, we break those proteins down into separate amino acids. We absorb them, and then through anabolic reactions, we can then take those individual amino acids and start to build them into larger and larger and more complex proteins. Right. So, again, two different types of catabolic reactions exist, and there's a reason as to why we're going to be going into this kind of much detail. Really, we're not going into that much detail, right? But when we look at the movement of electrons, like for instance, when we look here at oxidation, all right. Think of the term oil rig. I'm going to try and draw it out. Oil. Let's draw a capital L. I think this is actually neater than my own handwriting. I'll take it. Oil rig. So, again, I'll just kind of abbreviate oxidation. And then lose. Right. So think of the term oil rig. I'll kind of do a little thingy through here. Oxidation, lose electrons. And next slide, we'll look at where the rig comes into play. Right. But oxidation deals with losing or the loss of electrons. And the reason that's important is the electron is what is going to, again, at the end of the line, right? I'm talking like the last step where we generate massive numbers of ATP. It is all based upon the presence of electrons, right? One of the things with visible light, right? Take an incandescent light bulb, old school. You look at the filament inside, right? Many people, some of you guys don't know the horror in having an incandescent light bulb. After it's been on for 3 or 4 hours, it just blows, and you're like, oh, I gotta take it out. You're like, well, all right, you either do one of two things. You take your sleeve and you slide it all the way up over top of your hand to unscrew this light bulb because it's the temperature of the sun, or you lick your fingertips and just pray to God you're that fast. But then if it gets stuck, you're just you're just screwed. But anyways, there's a filament inside of that. What actually happens, what produces the light? You put up, I'm going to cliff notes this, but you flip on the switch and the current, the voltage, electricity, goes into the light bulb, into the filament, and essentially what happens is the electrons start to dance around. As the electrons dance around, that then emits the visible light. So it has to do with the movement of the electrons, right, as they're jumping back and forth from the shells. That kind of same idea, that same exact concept, right? It's the movement of the electrons which allows our body to harness and generate energy in the form of ATP, right? Because when we perform for instance, a muscle movement, right, or even the secretion of a hormone or the production of a hormone, all that takes energy. It's the actual energy which is stored in the bond, right, which has to do with the electron. When we will get to that, not today, but we will get to that. But it's the movement of the electron which is what's going to dictate and cause or dictate how much energy is actually produced. The other form of catabolic reaction we're going to look at, something called hydrolysis. So here's maltose, right over here. Hey, maltose. Okay. Maltose is just 2 molecules of glucose. So maltose is a disaccharide, right? Maltose is a disaccharide. There's no way in hell I'm writing out saccharide. All you're getting is di. Right? Disaccharide. Glucose is a monosaccharide. So if I take a monosaccharide and if I add together another, I'll just write M, another monosaccharide. So mono plus mono equals a disaccharide, or a 2-sugared molecule. If those 2 monosaccharides are glucose and glucose, when they combine, that's what then gives me maltose. All right. One of the ways I can break this up, if I look at the water molecule, if I think of it, if I were to say hydro water lysis breakdown, okay. So if I add a water to this, right, this water molecule, right, there was maltose plus a water. If I take that water molecule and if I add it, right, now in this, for instance, let's say this is the dividing line. Okay. On the left side of the reaction, here's the right side. That's pretty awful. On the left side of the reaction, right, we see, oh, there's a maltose molecule plus a water. When we come over here to the right side, we don't see that water anymore. Now you might say, well, wait a second, I see the HHO, which H2O. Okay, that's fine. But you no longer see a water molecule. They've been separated. Right? Part of the hydroxide, or part of the OH has joined this glucose molecule, and this hydrogen has joined this glucose molecule. So the water is no longer present. So by the addition of water, we've broken apart, right, the maltose into a glucose plus a glucose. Here, these are the forms that we're going to talk about, the form of anabolic reactions. Right, so on the previous slide, right, I wrote an oil rig. Right. On the previous slide, right, oxidation dealt with the loss of an electron. Reduction deals in the gaining of electrons. Right, so when we look, right, I didn't kind of make this point quite as well as I should have in the previous slide, so luckily I'll make it here. When we look, for instance, at the copper here, and if we look kind of more specifically right here, this is 2 plus. Right. Let's say if I then travel this way. What the hell? There's no more 2 plus. Huh. Where'd it go? Oh, if we look at that. Right? the 2 plus has now joined the magnesium. And why did that happen? It has to do with the movement of the electrons. Okay. So you guys remember there's protons, there's neutrons, and there's electrons. Protons, positively charged. Neutrons are neutral. They're like, yeah, whatever. I'm just hanging out. Electrons are negatively charged. Protons, the number of protons cannot change. Period. Right? Protons dictate what I am. Right? If the number of protons change, then what I am changes. Right? So, for instance, right, when we look at copper, right? Copper has, oh God, copper has, I should have looked this up. Copper has 29 with 95% certainty. Copper has 29 protons. Again, you do not have to know how many protons these molecules have. Not at all. Right? You do not need to know how many protons the elements have. No. But my point is, copper has 29. Yeah, it has 29 protons. That cannot change. If it does change, then I am no longer dealing with copper. Right? If it goes to 28 protons, then I'm dealing with whatever the hell I'm dealing with. Then I'm dealing with nickel. Right? So something entirely different. So, alright, so protons can't change. Neutrons, neutrons change. When neutrons change, it really is not terribly problematic. It can be, absolutely, right? But there are certain elements which just naturally, the number of neutrons are going to change, right? If you've ever heard of carbon dating, right? Right, with carbon dating, that's one of the ways that they can date. For instance, how do we know this dinosaur bone is 50 million years old? Or how do we know this human skeleton is 100,000 years old? Right? That's through carbon dating. Right? When a particular element has different numbers of neutrons, those are called isotopes. Right? They're different forms of the same element. So when you look at the different ratios and percent, there's a whole algorithm, right? When you look at the different percentages of the different isotopes, that gives you an idea that, oh, this bone is X amount old. Right? So neutrons, that doesn't change the charge of the cell. That leads us to the one and only thing that matters. And that is the electron. Right? The electrons can jump ship, and they do no problem. Right? Electrons are little, well, never mind. I'll keep it PC. But electrons, they have no issue jumping ship. Right? That's where, again, we see the copper, specifically the 2 plus, here. Well, it's no longer on the copper, but now it's on the magnesium, because it went from the copper to the magnesium. Right? The magnesium here, it was just, it didn't have anything. Now it's 2 plus. All right, we're going to talk about, next slide, we're going to talk about that more, so don't worry. Here, dehydration. So when you dehydrate something, oh man, you just suck the moisture right out of it. My mother, I don't think she's trying to create jerky turkey during Thanksgiving. I don't think she's trying to dehydrate it. But I tell you what, she does a pretty damn good, representation of what a dehydration synthesis reaction is. Because my God Almighty, that's the bird. It's delicious. The bird she cooks everything's wonderful. You just got to add, with each bite of turkey, you got to take a shot or two of just gravy, because there's nothing. Your esophagus doesn't. Your salivary glands can't produce enough saliva. Your esophageal glands can't produce enough mucus to supersede how dry this turkey is. But I love my mother. She's fantastic. She makes a mean apple pie. But to my point, right? A dehydration synthesis. So whereas the previous slide, here, we had a disaccharide. Well, if we added a water, it broke it into two separate monosaccharides. Here, if we remove a water from the glucose, right? If I dehydrate it, remove the moisture, remove the water, on this side of the reaction, we have the maltose plus the water. So that's an example of a dehydration synthesis. Now, this slide here, right? Oxidation reduction reactions. The previous slides, oh, here I just had oxidation. Here I had reduction. Right. They can never occur independently. Right? One of the first laws, right? Or not one of, but it is the first, right? Law of thermodynamics, right? The conservation of energy. Energy, there's a finite amount of energy in the world. It can be, you can't create it, you can't destroy it, you can like transfer, you can transform the form it's in, you can transfer where it is, but you can't create it nor destroy it. All right, so if this copper has a charge of +2, and here it doesn't have a charge, here it's neutral, that +2 had to go somewhere. Where did it go? All right. What did the copper ions gain? Protons, neutrons, electrons. Electrons are negatively charged. Protons are positively charged. So here, if copper went from 2+ to neutral, that means it became more negative, right? I'm +2, right? I am 2+. I'm +2. I can either, right? If I want to become more negative, I can do one of two things. I can get rid of my positivity. Well, we can't do that. Protons are positive. I'm still copper, so I know that's not it. That can't be it. Well, what if I am copper 2+ and I add 2 electrons? That's supposed to be a 2. Just for the point of it. Oh my god. I'm just going to leave that to 2, right? If we add 2 electrons to this 2+ copper, all right, well, if I add 2 negatives, to 2 positives, they cancel out. Now they're neutral. That is why we see copper go from 2+ here to becoming neutral here. Those electrons had to go somewhere. They can't just be floating in the ether. Something had to pick them up. All right, so for this example, magnesium, we see right here. I was neutral. I'm magnesium. I'm like, listen, don't mess with me. I'm good. I got no problems at all. I'm just neutral. If we follow over here, we see that magnesium suddenly now becomes +2. So what had to happen? Did it pick up 2 protons? No. It's still magnesium. So it still has the same number of protons. Did it pick up electrons? Well, if it picked up electrons, it'd be more negative, right? Because electrons are negatively charged. So here the magnesium went from neutral, I went from no charge at all, and I went to just super positive. Like, hey, everything's going to be fine. So I got rid of a bunch of negativity, specifically 2. So this magnesium picked up 2 electrons, right? I promise that is as chemistry heavy as we're getting, right? Again, chemistry of the body. There's all chapter 2 stuff, right? But this is, it's been so long since we've kind of talked about this, reviewed this stuff. I had to kind of, excuse me, had to talk about it because of how important this is at the end when we get into something called the electron transport chain. Who knew electrons would be important for something called the electron transport chain? So this here, right, the first thing we're going to start to get into is when we talk about carbohydrate metabolism, right? How is it we get that carbohydrate into the actual body? Great. So it doesn't matter what you've eaten, rice, pasta, whole grain rice, whole grain bread, whatever vegetable, whatever fruit you want to name, doesn't matter, right? Pretty much, right? And for the most part, right? Anything that wasn't at one point living, right? When I say living, I'm not talking about plants. I mean like making a noise, right? Breathing air, right? It's a carbohydrate. There are some exceptions, but by and large, right? That's why when we talk about carbohydrates, generally people think about like breads and pastas, which is correct, but they always forget about like fruits and vegetables, right? So, anyways, this molecule here, let's just call it some complex carbohydrate. Oh, awesome! You ate a whole grain fiber, or you ate some whole grain. You ate a whole grain hamburger bun. Awesome. Those polysaccharides, those large chain sugars, have to be broken down, and they are. They're broken down to disaccharides, and then eventually those disaccharides are broken down into monosaccharides. Awesome. So we talked about the enzymes, right? Maltase, for instance, lactase, for instance, here in the brush border. Remember this being the brush border here. I had mentioned how there's via active transport for glucose and galactose and via facilitated diffusion for fructose. But there's something called there's different types of GLUT transporters. We're not going to talk about which ones are which. But there's something called a GLUT transporter, which grabs a hold of the monosaccharide here and carries it through the intestinal wall and eventually absorbs it into the bloodstream. Right? That's what's going to occur via carbohydrate metabolism. Now we see, regardless of the type of carbohydrate, nearly all will end up as glucose. So whether it's maltose, fructose, sucrose, lactose, etc., whether it's a milk sugar, right? Lactose is a milk sugar. You see it in milk. Period. It's the only place you see it. Right. So whether it's lactose, right, which is a disaccharide, which is a glucose molecule plus a galactose molecule. Whether it's maltose, which is 2 glucose molecules. Whether it's fructose, right? Whether it's sucrose, which is 50-50 glucose and fructose. Right, even these large chain, these polysaccharides, these large, 100,000 chain saccharides, they too are going to be broken down into glucose. Nearly all of them. Right, we see a very small amount of glucose is in the blood at a given time. It's a range between, right around 5 grams. Right, it's a little closer to 8 grams, but it's between 5 to 10 grams. Right, so call it 5, right, but the weight of a nickel. We see when or why might there be more or less in the blood? Oh, all right, so, if you're just eating food, there's going to be more glucose in the blood. Just makes sense, right? We just ate food, regardless if it was pasta, cheeseburger with bacon, extra sharp, preferably Vermont sharp, onions, mushrooms, tomato, some mayonnaise, a little bit of mustard, sweet maybe. Whew. So regardless though, milkshake, cheeseburger, fries, pasta, kale, kale, whatever it was, right? Your glucose levels are going to rise, right? Your blood glucose levels are going to rise. All right, we can all understand that. But what else? What else might your glucose, specifically your blood glucose levels, when else might they elevate? Right, take 10 seconds, think about it. How about when we're exercising? Right? When you're exercising, right, there's small amounts of release of epinephrine, right? That's going to stimulate the release of glycogen from glycogen stores, elevate blood glucose levels, right? In times of even acute, even chronic, especially acute stress, when cortisol levels are elevated, that too is going to elevate glucose levels, right? So there are certain times where it's okay to have elevated levels of glucose. It's normal to have elevated levels of glucose, right, at certain periods of time. For instance, post-consumption, right, exercising. The problem is when it's sustained elevated. That's the problem, right? Well, that's kind of what we'll get into. So we see hormonal effects of activity and food intake function to regulate glucose levels. Yeah, we've just eaten food, right? Depending on what it is. As your glucose levels elevate, that is going to in turn cause what hormone to be released from the pancreas? Insulin. As blood glucose levels rise, so I just ate some pasta, I just ate some white bread, right? I just ate a piece of cheese, I just ate some pepperoni, I just ate some bologna, whatever. I ate a banana, right? Ate some kale. Ugh. Right? My blood glucose levels are going to rise. Now, all of the things that I just mentioned, right? The image of the desserts on the page, absolutely all of those things are going to impact your blood glucose levels differently, right? This thing, I know what the heck these are, right? These desserts on the slide, those are obviously going to drastically impact your blood glucose levels, way more than even rice, cheese, pepperoni, etc. Way more than those things would, right? Because these, right? If I handed you a piece of paper and said, "Hey, turn this into some origami, or turn this into a paper airplane. I need you to turn this into something you can throw." And all I did was hand you a piece of paper, right? Something that you can throw and glide. And all I did was hand you a piece of paper. You have to take that piece of paper and then actually turn it into an airplane and then throw it. You have to fold the piece of paper 8 or 10 different ways. You have to actually utilize energy to do it. You actually have to take time to make the energy, or to make the airplane. Right? That's what consuming, for instance, whole grains, or consuming, pepperoni or bologna, something that has a lower glycemic index. Right? It takes a greater amount of energy. It takes a greater amount of time for your body to absorb, right, that glucose. Much different than this dessert. That's me handing you a paper airplane already made and say, "Hey, throw this thing which glides." You can do it immediately. Right? As soon as I hand it to you, you can immediately throw it and it glides. That's glucose in the form of, right, table sugar, fructose, right, within these desserts. As soon as you ingest it, right, your body begins to, as soon as it hits the intestines, your body begins to rapidly absorb this stuff because it is already in its most simplified form, right? There's monosaccharides. They don't get simpler than that, right? So therefore, the haste, or the speed in which these monosaccharides are absorbed are significantly higher than amino acids, fatty acids, even complex carbohydrates. So we see, why is this so important? Why do we care about glucose regulation? Right? Why? Like, we all know, okay, we got to breathe. Okay, we got to eat stuff. But, like, why? Right? It's the question of, like, why shouldn't I go out to my car and just start it up and turn it off repeatedly? Is it a bad thing? Yeah, it is. Why? I have no idea. I have some theories, but I really don't actually know the reason why I shouldn't do that. I just know it's a bad idea. It's kind of the same thing with the body. Right? We all know, you gotta get food. We gotta eat food. We gotta breathe air. We gotta breathe oxygen. Because if we don't breathe oxygen, we're gonna die. But then if you're like, well, why? Why is oxygen important? It's like, I don't actually know. Right? That's kind of what we're going to get into. Right? Glucose regulation. Right? In order for our body, for instance, in order for our skeletal muscle to move, right? In order for all the different sphincters of our body to function, in order for our glands and our organs to produce digestive enzymes, and for our cells to produce different hormones, all of these things require ATP, and they require massive amounts of these things. I mean, on a daily basis, an absolutely astronomical figure of ATP that is produced on a daily basis. And they're produced via what's called anaerobic and aerobic respiration. Right? Anaerobic means without oxygen. Aerobic means with oxygen. Right? And we are going to break down carbohydrates. We're going to break down proteins. We're going to break down fats. Right now, depending on your energy level, depending on how, what the demand is for the ATP, that's going to dictate, well, what's the source of this, or what source are we utilizing to generate this ATP from? Right. The bulk, right? The vast majority of ATP that you utilize, or that you burn, or that you produce, that you produce on a daily basis, are generated from the breakdown of fatty acids. Even if you are a world-class sprinter, right? The bulk of ATP you produce are from fats and fatty acids. However, if you are a sprinter, you are going to produce a much greater number of ATP from carbohydrates than you would, from somebody who wasn't a sprinter. But in combination, or in comparison-wise, even that sprinter still utilizes a greater percentage of fats to produce the ATP. Right? It's just kind of like filling up sticking a garden hose in the ocean. Right? Stick a fire hose into the ocean and turn it on. Right? That fire hose is always going to be running. That fire hose is always going to be pumping out, 50 gallons a minute. If you then take a cup and grab a cup of water and dump it into the ocean, that's ATP production from carbohydrates, right? That's the ATP production from non-fatty acid sources. It's a small amount, but if you take and just continuously dump more cups into the ocean, you're continuously producing greater ATP from carbohydrates. However, the bulk is from that garden hose. The bulk is from the fatty acid source. Right. There's a couple other things: ketone bodies, creatine phosphate. Those are some other sources from which we can derive ATP. We'll talk about those more. So we won't get to it today. Probably not next. Well, we might get to it next class as well. But we're definitely not going to get to it today. But we will. These 4 steps here, right? This is again simplified, right? Kind of Cliff Notes version of the 4 different steps. Right? We can see glucose, right? Pyruvic acid, right? Acetyl-CoA. Way over here, right? The electron transport chain. You see, remember the electrons from, right, the 10-15 minutes ago, right? This is the actual step for which those electrons are important, right? Is right over here. But before we're able to get to that electron transport chain, we have all of these steps here that we have to get through. And you can't just I'm just going to go ahead and skip this one and go from one to... Nope, that doesn't happen. Can't do that. Right? There's things produced along the way which are necessary for the subsequent step. Right? Through glycolysis, that leads to something called pyruvic acid. Pyruvic acid has to be created so we can then produce something called acetyl-CoA. Acetyl-CoA is necessary because that has to plug into the Krebs cycle. The Krebs cycle is important because there's a number of other coenzymes, or when I say coenzymes, other molecules which are necessary so they can plug into the electron transport chain. So each step has to occur. We can't just go from 1 to 2, and then from 2 to 4. Nope, that can't happen. Right? You've got to go through each step of the process. As far as anaerobic and aerobic, none, aerobic, oxygen. So without the presence of oxygen, glycolysis. The first step in ATP production is glycolysis. Now, we do produce a small amount of ATP via glycolysis. It's a very small amount. Right? 2, to be exact. Technically, we produce 4, but it actually costs 2 to generate the 2, so there's a gain of 2 ATP. That's it. Electron transport chain, oh my god, we're producing like 14 times that. Right, so we're talking about a significantly greater amount of ATP molecules. But again, glycolysis, this is that first step in converting that glucose into something called pyruvic acid, or something called pyruvate. Aerobic respiration, again, that does not require oxygen. And this is something which occurs in the cytoplasm. Remember, the cytoplasm, that was, again, we're going back to chapter 3, but the cytoplasm, that was the kind of liquid portion of the cell and all of the organelles within it, between the plasma membrane and the nucleus. That was the cytoplasm. That's where the aerobic respiration occurs. Right. Aerobic respiration, that specifically is going to occur in the mitochondria. Right? Within the actual mitochondria itself, there's different quote-unquote chambers, membranes, there's a labyrinth, there's layers in which the actual electrons can mobilize and move, which is going to help generate the ATP. At the end of the line, when we get to the electron transport chain, we will look at that. So here, first step, glycolysis. Awesome. You see net of 2 ATP which are produced. Yeah, it costs 4, right? Or I'm sorry, we technically generated 4, but it costs 2. So there's a net of 2 ATP which are produced. Now we see it produces coenzymes NADH and as well as a hydrogen. We see middleman carrying energy to subsequent step. One of the things that's important, hydrogen is positively charged. What's one of the things that we talked about with electron? Ooh, electron's negatively charged. What happens when you hook up a negative and a positive? Think of a magnet, right? They attract, right? So the movement of this hydrogen, we're going to find out, is what actually deals with the movement of this electron. All right. The other thing, production of pyruvate. Right. Eventually, pyruvate, right, again, this guy here. Eventually, pyruvate is going to enter into the mitochondria and become acetyl-CoA. But glycolysis, that is the anaerobic step. That is anaerobic, does not require any oxygen. So we are in the cytoplasm. That's where we are, up here. We're not in this yet. Right, we're not in that yet. We are up here in glycolysis. All right. So we generate the pyruvic acid or the pyruvate. Those are the same thing. All right. So is it all hunky-dory? Hey, great. Once pyruvic acid and pyruvate, once that's generated, that's going to then immediately dump into the mitochondria. That's dependent upon one particular thing. That's dependent upon O2 levels or oxygen levels. With adequate oxygen levels, pyruvic acid will enter into the mitochondria. Awesome. In inadequate oxygen levels, pyruvic acid gives rise to something called lactic acid. And it works out better if I don't actually cover half the damn word. Produces something called lactic acid. Lactic acid, lactate, right? Again, same thing. Lactic acid. This is one of the byproducts of energy production, specifically through glycolysis, right? With glucose. Think of it this way: because of glycolysis and how inefficient it is. All right, with where we live, with all of the number of tractor trailers that we see, the number of hills that are on the interstate. If you've seen an older tractor trailer driving up a hill, and it starts to bog down, but you see like these just billows of smoke pouring out of the smokestacks. And you look over to a new tractor trailer, you're like, oh, the thing's not like pouring out any smoke. It's because that newer tractor trailer, that newer truck is burning the fuel as efficiently as that engine possibly can. The older the engine is, the more inefficient it functions for a bunch of different reasons. So there's more waste product. You can see it by the smoke which is billowing out of the smokestacks. Right? Same idea here. Because of the inefficiency of glycolysis, there's a byproduct. Right? It's called lactic acid. Right? So when we produce energy, right, or ATP, right, when we undergo a great deal of glycolysis, like in those activities which are very quick, like sprinting, for instance, right, something which is very immediate burst of energy but very short duration, those sorts of things. That's what's going to, right, when we talk about aerobic exercising or anaerobic exercising. Anaerobic exercise is lifting weights, right? It's anaerobic. Lifting weights is a very short duration activity. You're like, well, I lifted for 3 hours. Yeah, but the actual lifting of a weight, there's a reason as to why you do it. Oh, you do so many in a set. Oh, you do 10 reps per set, and you do 3 sets with a rest period in between. There's a reason as to why it's built like that. Right? We'll talk about that a little bit later on. Right? But there's reasons as to why. Again, muscle soreness, muscle fatigue, right? One of the theories, and it's kind of an older theory. It's been disproven, but the idea that lactic acid is actually what is causing muscle cramping, muscle soreness. That's actually not the case. We're not entirely sure actually what it is that's causing the muscle to be exceedingly sore. A few different theories, but one of the things that we know, it's not the excess of lactic acid buildup. So here, right, within the mitochondria, we begin aerobic respiration, which requires what molecule? All right, it's aerobic, so it requires oxygen. Awesome. But it also requires pyruvate, because pyruvate is eventually going to be transformed into acetyl-CoA. Down here, right, coenzyme A, acetyl-CoA, acetyl-coenzyme A, those are all of the same thing. It's got 3 different names. Sorry. So again, right, what if there's a limited amount of oxygen present? Again, we just talked about what happens to the presence of lactic acid buildup. But one of the things with lactic acid, it's not just like, all right, it builds up and everything's hunky-dory. One of the things that happens, lactic acid is shunted to the liver. There's something called the Cori cycle, and that liver then converts that lactic acid and recycles it and shunts it back into glycolysis. Right? And no, we won't get into it today. There's a graphic I have where what we can actually see with the Cori cycle. We can actually see the movement of this of the lactic acid to the liver and the conversion back into an effective, actual usable form of, in glycolysis. Here, right, where pyruvate is actually converted into acetyl-CoA. Right. We see the pyruvate enters into the mitochondria, right? And this right here, right, we all know, when we breathe oxygen, awesome, and we breathe off carbon dioxide, right? But like, where does that come from? Right? Like, all right, is it just there? Like, where's the carbon dioxide actually come from? This is the first step where some of the carbon dioxide is generated. Right? We see when pyruvate is actually converted into acetyl-CoA, that actually generates a CO2 molecule. Right? So this is why like CO2 will accumulate incredibly quickly inside of the body. I'd mentioned we are producing astronomical figures of ATP. Well, in order to do that, pyruvate has to be converted into coenzyme A. In order to do that, one of the byproducts is the generation of CO2. Hence the reason why CO2 will always be produced inside of the body. Has no choice. It's one of the byproducts. Good. Why that's important? Again, lastly, acetyl-CoA. Again, this guy here. Acetyl-CoA is necessary because this is plugged into the third step, into what's called the Krebs cycle. Now, the Krebs cycle, also called the TCA cycle, also called the citric acid cycle. Again, TCA, citric acid cycle. Right. But this is the thing with the mitochondria. Right? How I'd mentioned. No, first off, you do not need to memorize every step in this cycle. No. I'm not proud of this. I used to be able to every amino acid, every enzyme. Like I said, I'm not proud. A lot of things in life I'm not proud of. That's one of them. But the acetyl-CoA, right? This is where it went to, right? Okay, so it was produced here in the third step, in the second step, and it's plugged into the Krebs cycle. Now, when I say it's plugged into, like, one of the things we always kind of get confused with, and this is the last slide I want to cover because I'm already about over time, is, all right, well, what about the oxaloacetate or the citrate or the isocitrate or the alpha-ketoglutarate? Like, where are these just hanging out? Like, what? I don't get, like, sometimes we just get confused. Like, where do these different amino acids come from? Like, are they already there? Like, what's going on? And there's a number of processes, right, where all it takes is the addition or the removal of a molecule, and that process halts. Right? Imagine this. If I drew this arrow here, this arrow here, this arrow here, I drew this arrow here. Right? So, think about it. This arrow leads to this arrow, which leads to this arrow, which leads to this arrow, and then so on and so forth. That's what we have here. Awesome. Imagine acetyl-CoA is this arrow Let's imagine acetyl-CoA. Let's just imagine acetyl-CoA is that arrow right there. If I remove acetyl-CoA, are the other arrows still there? Yes. Just like down here. Citrate, isocitrate, alpha-ketoglutarate, so on. Are all of those still there? Yes. But does this arrow here, right, does this arrow here, does it continue on? No, it doesn't, because it can't. Because it's missing the, for instance, missing this block, if you will. That's exactly what happens here. The acetyl-CoA causes the oxaloacetate, it mixes with the citrate synthase to give rise to citrate. Without the acetyl-CoA, this step here does not happen. Right, does that make sense? Right, so it has to do with the fact that acetyl-CoA plugs in and then citrate synthase causes that conversion from acetyl-CoA to citrate. So I plug acetyl-CoA back in. Oh, guess what? And the cycle continues. Now let's just say, hey, you know what? I just ate a ton of food, or I was exercising, I am no longer. So I don't need to produce as much ATP as I once did. Now we can remove acetyl-CoA from the Krebs cycle. And if we remove acetyl-CoA from the Krebs cycle, we no longer produce, again, the citrate. Right. Next presentation, we're going to look at how that's one of the things that can occur is even as acetyl-CoA gets plugged in to the Krebs cycle, as it gets plugged in, let's imagine if our energy production goes down, we don't need to produce any more ATP, or not anymore. We will always need to produce massive amounts of ATP. But instead of producing at 100% capacity, right, we only need to produce at 90% capacity. So that means we can start to store some of this excess ATP. Citrate can actually get kicked out of the mitochondria. Right? We will talk about that next presentation. So if you, at this point, right, if you guys have any questions, please let me know. Right, next class, right, we'll get into the electron transport chain and don't go into anaphylaxis. We will talk about certain steps of this slide, and we'll get into kind of all the rest of these, all the rest of, glycogen storage, adipose tissue storage, adipose tissue production, and all that kind of fun stuff. So until then, everybody take care, and we will talk to you again on Thursday.